Hello students. So our next topic is about a gene. Before starting with transcription and translation, we should know the basic structure of a gene. So find structure of the gene. A gene, it can be defined as a segment of DNA which carries the biological information for the formation of some RNA molecule that can be non-coding genes or coding genes. So genes can be of two types: non-coding genes which does not code for any protein, and coding genes which will code for some proteins. So these genes, they are a segment of DNA that will carry the information in it. Next, some of the examples of non-coding genes are rRNA, that is ribosomal RNA, tRNA, that is transfer RNA, and microRNA genes. So these genes are non-coding genes. They will not code for any protein, but they will help in such process. Genes may vary greatly in size, but the average size of a gene is about 1000 base pairs. So the average size will be 1000 base pairs. Base pairs were the ATGCs. Next, each gene coding region, it carries a promoter. Promoter is the controlling element that is present at the upstream region and that drives the expression of a gene. So suppose this is a gene, okay, this is a gene, this is segment of a gene, and here the promoter region is this at the top, at the upstream. So this side is known as upstream, and we also call it as promoter site. And a terminator. So a terminator that is present at the downstream region of the gene and is involved in the termination of transcription. So when we have to stop the transcription, this terminator will act. It is present in the downstream. See here, this is the terminator region, terminator site. So this is the downstream site. This is our upstream where promoter site was present. This is our downstream where terminator site is present. And this terminator region is responsible for termination or stopping the transcription process. Transcription was conversion of DNA into RNA. So that process will terminate, will stop at this terminator region of the gene, which is present in downstream region. So genes are separated from one another by intergenic region on the DNA molecule. So one gene is separated from one another by intergenic region. In case of virus, genes are closely arranged with very little intergenic DNA between them. So in virus, the intergenic region is very less. But in higher organisms, the genes are more spread out and are usually separated by very long intergenic region. So in higher organism, the intergenic region is long. This intergenic is also called as spacer. It is the gap, right? It is giving the space between one gene, two genes. Next, the total number of genes in different organisms also varies from few thousand to several thousand. For example, in case of E. coli is a bacteria, single chromosome carries approximately 4,500 genes. So in E. coli, single chromosome, it carries approximately this much genes. Whereas in yeast, which contain 16 chromosome, okay, in yeast we have 16 chromosome, it contains about 6,500 genes. So the total number of genes present in an organism, it varies. In human also it varies. Next, it has been estimated that human genome would contain about 20,000 to 25,000 protein coding genes. So we have approximately 20,000 to 25,000 protein coding genes in us. Okay, and this is suggested that only 1 or 2% of the human genome codes for protein. Only 1 and 2% out of this will code for a protein, and the rest include the genes for different type of RNA molecules and several kind of DNA repeats. So only 1 or 2% will code for some protein in human, and rest all are involved for other purpose. So this data you don't have to remember. For human genome, you can, since this is again there in your class 12 also, biology part, you will study about this human genome. Next, we'll study about introns and exons. So let us see what is this. Gene may consist of stretches of intervening DNA sequences which do not carry any message and are called as introns. So simply you can say the genes, the DNA sequence or the genes that do not carry any message are called as introns. Along with this, the region that contain biological information are called as exons. So introns, it does not code for anything, it does not carry any message, but the exons, it code for a protein. So it contains the biological information. So in exon, we will have the biological information, and introns, it does not carry any message. And these two introns along with exons, such genes are known as split genes. And it is also called as discontinuous gene, because we will have introns, then exons, then introns, then exons, like that. So this is known as discontinuous genes, or you can also say mosaic genes, and are common in higher organisms including humans also. In us also we have a sequence of exons, then introns, then exons, and then introns. I'll show you the diagram. See here, suppose this yellow color, this is intron, then we have this green one, this is exon. Again we have intron and then exon. So this intron, it does not carry any message, and the biological information is contained in exon. So the number of introns in a gene may vary from none to more than 100. So the number of introns may vary. Organisms having a mechanism to remove introns after transcription of a gene. So this process is called as RNA splicing. See here students, introns are not needed since it does not carry any message, any information. We don't need this part. We only need the exon, which carry the biological information, right? So these introns, can you see here? These introns are removed from the gene, and this process, the removal of introns, is known as splicing. This process is called as splicing. After removing this exon will then join. This will then join. This we will learn in transcription also. So this process is known as RNA splicing. Now prokaryotes, they do not contain introns in their genes. Therefore, the splicing mechanism is absent in such organisms. Since prokaryotes, it does not contain any introns, so splicing will not occur in them. But in us, splicing used to take place. Genes may occur in eukaryotes. You can say that splicing, RNA splicing occurs. Genes may occur singly or in cluster. The genes that are present in cluster are related, and two common examples are the operons present in bacteria. So operon is present in bacteria. and the multi-gene family in plants and humans, animals also. Some genes may lose the biological information they had once coded for as a result of mutation. See, mutations might occur in a gene, and if mutation occur, they might lose that information which they contain. Okay, so that they are no longer functional. So such gene may become non-functional, and such genes are known as pseudogenes. If they lose the biological information they had once coded for because of mutation, they may become non-functional, and such genes are called as pseudogenes. And mutations may occur because of so many reasons. We will study about mutations.